all, what is embryology? What we study in embryology, we should be aware about that. In embryology, the study of embryo formation, embryo development, all this. You know how embryo formation by zygote, by fertilization. Let me take a whiteboard. Right. So in embryology, we study about embryo formation and development. Apart from the embryo, the other development of endosperm also formed. This endosperm also we discussed in this embryology. So first of all, embryo developed from the zygote. All we know from zygote, which is diploid condition. So this embryo development from fertilization, right? So what is fertilization? Fertilization means fusion of male plus female gamete. The both male and female gamete fused. As a result, zygote formed. The single cell diploid zygote further developed into diploid embryo. Apart from the yellow, we will discuss later in double fertilization and sorry for inconvenience. Right. Endosperm also developed. Apart from them, we will discuss later how the endosperm formed, how the embryo formed in double fertilization process. So anyway, what is embryology? Embryology, we study formation and development of embryo. Okay, that's okay. The embryo development from the diploid zygote, which is unicell, single cell, and the zygote development from the male gamete and female gamete, right? How the male gamete developed? How the male gamete developed? Here the male gamete developed from the microspore or pollen grain, microspores by microspore or pollen grain, or another name is pollen grain. Here the female gamete developed from the, here the microspore male gamete and female gamete development from the megaspore. So here the microspore and megaspore, right? Okay, done. Here the development of the microspore inside microsporangium, which is called as anther. Microsporangium, or another name is called anther. And here you can assume this microsporangium. And what about megaspore? Hello? Megaspore. Here the megaspore development inside the megasporangium, and which is called as ovule. Development inside the ovule. Right? So development inside the ovule, right? Now the microspore or anther, microsporangium anther. This microsporangium anther, these both are parts of stamen. And here the ovule, the parts of gynoecium, stamen. These both are parts of here the gynoecium and stamen. They both are the parts of flower. So what we discussed here. So after formation embryo development to seed development as a plant. Okay, plant. After plant formation, the plant's parts also develop, such as root system, shoot system, and leaf. When the vegetative development occurs, next it is further developed as a, that is, when it is reaches maturity stage, in maturity stage, reproductive organs are developed. The reproductive organ in angiosperm is flowers, right? So what are the flower parts? Calyx, corolla, androecium, gynoecium, right? In androecium, group of stamens, gynoecium, in gynoecium, ovule present. In group of stamens, the stamen part microsporangium or anther. In ovule, megasporangium, which is developed as a megaspore, and anther developed as a microspore. This microspore further developed into female gametophyte and further developed into female gamete. As a similar way, here also, megasporangium, megaspore, female gametophyte develop, which is produce female gamete. So, as we discussed earlier, the fusion of male and female gamete fertilization process, zygote formed, zygote developed into embryo. So, this is the cycle. So, in this embryology, we discuss each and every one. Each and every one means here the microsporangium structure, how the microspores are production. This is called as a microsporogenesis, and megasporangium structure. After that, megaspore production. This process called as a megasporogenesis. So after microsporogenesis, this microspore developed into male gametophyte. Then the next one, the development of male gametophyte, one unit, and another one, the development of female gametophyte. So anyway, after developed male and female gametophyte, after that, here the another one, before fertilization, what happened? Actually, the pollen grain transferred to one place to another place, right? Then only they can fertilize each other. So this process, this process means here, so after full development of the, here the male gametophyte and female gametophyte, which are traveled one place to another place, this process called as pollination. And this in embryology topics, we discussed about pollination also. So pollination also we need to discuss here. After pollination, what happened? The pollination means a simple pollen grains transfer one place to the another place and reach the stigma of the gynoecium. If the transfer is facilitated by wind, which is called anemophily, or insect, birds, ornithophilia. So whatever it may, and water and rain drops, these are all involved as a vector to transfer the mature pollen grain from one place to the another place, right? After that, when reach the gynoecium of stigma, there is a small interaction between the pollen grains and the stigma. After interaction, they recognize the proper pollen grain. It is the same pollen grain, accept the same species pollen grain, and reject the another pollen grains. If it is what is here, actually these are the very sensitive. The stigma region very sensitive to the foreign material. Only this is accept this own material. The own pollen grain they can accept. The another pollen grain, the foreign pollen grain, they can reject. So this process called as interaction. After interaction, the pollen tube develop this from through pollen tube. For male gametes are developed. Male gametes reach to the ovule inside the female gamete. So all this process occurs in this embryology. So let's discuss one by one. Let's start with microsporogenesis. Is it the first unit? Microsporogenesis and male gametophyte development. And second one, megasporogenesis, second unit, megasporogenesis and female gametophyte development. These two very, very important. In first block, the three topics. First unit, microsporogenesis, male gametophyte development, megasporogenesis, female gametophyte development. And next one, pollination types. That is third unit. So this is the first block. This is very, very easy and important one also. In this, you can select all questions, all topics. Already told you from beginning, there are 4 blocks. In each course, 4 blocks. So when we prepare the question paper, we balancing the each and every block. That's why my humble suggest you for your just if you want to just pass mark, just select any easy 2 block. Any easy 2 block so that you can easily pass. Right? Is it clear? Next. Let's enter into the first unit, that is microsporogenesis. Just wait. Let me take. Right. Microsporogenes. Let me take the PPT slide. Just wait. That's simple here. What is microsporogenesis means? Microsporogenesis means in the name itself given. Name itself given sporogenesis, which means, for example, just take another one. Right. So this is the stamen. These are the stamen. Just wait. Right. Already we discussed earlier stamens. The stamens, the part of the flower. So in flower, we take the stamens. If we take the stamens, there are 2 parts clearly, filament and anther. So this is the anther transfer section. If you take transfer section anther, there are the 4 loops, 1, 2, 3, 4 loops and 2 microsporangia. And these 4 loops. Inside the 4 loops, here the microsporangium. Here also microsporangium. Here also microsporangium. This also microsporangium. What happened in microsporangium? Actually, if you take in detail here, in this anther, there are clearly two types. One is in this anther, clear 2 types are wall, anther wall. Another one is sporogenous tissue. Sporogenous tissue. Anther wall, porogenous tissue. And inside the wall, here is the wall, again 4 layers. The 4 layers are epidermis, endothelial, middle layer, and last one tapetum. This is all from wall layers. And the next one, after wall layers, the next one sporogenous tissue. If we take the diagram, this is all together anther wall. So first layer epidermis, second elongated cell endothelial, and just small these cells, these are middle cells, middle layers. And the next one, this tapetum layer enlarged. And next part completely, let me change. Blue color sporogenous tissue. So this is in one part of the anther. Anther one lobe. Actually, I will clearly draw this lobe only. Only this lobe in detail, right? So after formation, this blue color sporogenous tissue, what happened? Let me draw clear here. The sporogenous further developed into uh, mother cell, actually archisporial cell. Archisporial cell further developed into microspore mother cell. Microspore mother cell. This one, this is called, this, which is diploid, and this is called microspore mother cell. After formation microspore mother cell, here the meiosis division takes place. After meiosis, the haploid spores are formed. These are the haploid spores. So the genesis, the production of the microspores are the pollen grain. Microspores are pollen grain. This process called as microsporogenesis. Now, where are the microspores or pollen grain? This pollen grain and microspores are present inside the anther, right? So later, so anyway, after formation, this microspore and this development also two type. One is successive development. Another one is simultaneous development. Uh, First, let's complete successive development. In successive development, first this is microspore mother cell, right? This one, microspore mother cell, MSM, microspore mother cell. First one, microspore mother cell. So this after microspore mother cell formation, actually here the meiosis division takes place. So you remember one thing, the meiosis is the two stages, meiosis 1 and meiosis 2. The first one, meiosis 1. After meiosis 1, what happened? This microspore mother cell, this is successive type. Successive type. In successive type, what happened? This 2n, 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 the divided into two haploid nuclei. After that, immediately formed septa. So this is first mitotic division. Next, second mitotic division, it is almost to mitotic, it is resembled to the mitotic division. What is mitotic and what is meiosis here? In the mitotic division, after mitotic division, the chromosome number neither decrease nor increase. In meiotic division, after division, the chromosome number completely decrease off. That's why here the microspore mother cell, 2n, this 2n microspore mother cell, after meiotic division, two haploid cells are formed. After formation, two haploid cell again divided into another meiotic division. So after another meiotic division, which is this two becomes into 4. 2 becomes into 4. Like this. Sorry. Next, another septa form. So this is the first transfer sector. Again, which is these two nuclei also developed into second meiosis division. Again, another septa formed. So this is called successive division. This is called successive division type. Successive. This is called successive. This is called successive type. And let's take another one. Is it clear? Successive type clear? After successive type, and second one is simultaneous type. The simultaneous type means this same microspore mother cell, after meiotic division, first meiotic division, two nuclei formed, but these two nuclei does not form the septa. After formation, two nuclei, no septa formed. Here also no septa formed. What happened here? Just wait here. After second meiotic division, again two nuclei formed. Final. After formation of 4 haploid nuclei, immediately at a time, the septa formed. Transfer and vertical. So this is called simultaneous. Simultaneous type, the name itself given simultaneous. So anyway, the formation of the microspore and pollen grain tetrad, by two methods. One is successive, another one is simultaneous. In successive, first meiotic division, immediately it follows the septa formation. That means cytokinesis occur and septa formation immediately follows. And second, meiotic division, again another septa form. That is successive. In simultaneous, after meiosis 1 and 2, after formation, there is no formation septa after 1. Like successive, immediately when reach the final, that is meiosis 2, then only the septa formation between immediately. That means at a time the septa formation between the nuclei. Right? So this is the formation of microspores and microsporogenesis. And let's see the arrangement of microspore while they are formation. While their formation, there are different type of arrangement of the microspore. Let's see the different type of arrangement of microspores. Right. So here the first one, tetrahedral type. So what you don't forget the formation of whether it is successive type or simultaneous type, any type. Let's see, give here the first of all, sporogenous micro spore mother cell 2n meiosis 1, two nuclei, meiosis 2, another two nuclei formed. This is the whole concept, right? So let's see here. So but the formation different. Maybe it is a tetrahedral type or isobilateral type like this. This is isobilateral type, or decoset type, or linear type. Mainly, majorly 4 types of microsporogenesis occurs. That means pollen grains arrangement occurs. Let's discuss one by one. The first one, tetrahedral type. What happens in tetrahedral type? In tetrahedral type, after meiosis 1 and 2, whether it is successive or simultaneous, any type of manner, here the 4 tetrads are formed. But in one view, we can observe, we can see only 3 microspores. The rest of the microspores and behind the that backside the 3 pollen grain. So this type of arrangement called as tetrahedral arrangement. Say simple tetrahedral arrangement. Here the simple we can observe here one pollen grain, second pollen grain, third pollen grain. But backside third pollen grain also present, but which is not appear one plane. If you see backside, we can observe. So that is the tetrahedral type commonly occurs. The mostly 90% of the angiosperm, this type of arrangement occurs generally. And second one, isobilateral. We can see isobilateral, the pollen grain arrangement side by side. We can observe the all pollen grains in a view. In a plane, we can observe the 4 pollen grains. So this condition called as isobilateral. And second one, decoset. In decoset, the two pollen grains opposite direction. That means the first in decoset manner, these 2 opposite direction pollen grains. So remember, any pollen grain tetrad formation, 4 pollen grains are there, and rest of the 2 also opposite direction. In this opposite direction, this is this opposite direction. So that is called a decoset manner. The another one is linear type. If we see the linear type, the all are arranged in the single line. The 4 pollen grains, 1 and 2 and 3 and 4, they are all arranged on the single plane, single line. So this is called linear type. So anyway, we see one is the formation type, successive or simultaneous. Arrangement of pollen grains also we discussed. Right? Is it clear? Next one, after formation, any manner, any arrangement, finally it is to be developed as a male gametophyte. Let's see the male gametophyte development. Here, remember one thing, the microsporogenesis very, very important for short answer. And male gametophyte very, very important for long answer. Right? So now the short answer completed. Now let's see the male gametophyte development. If in examination point of view, if they ask about male gametophyte development, you can start with microsporogenesis until male gametes formed. Remember, in examination point of view, if they ask write about microsporogenesis, they simply write about the definition of microsporogenesis, the types of formation, successive, simultaneous, and types of arrangement. Simple. No need to write about the development of male gametophyte. Remember it. Right? Now let's see male gametophyte development. So anyway, after that, the microspores are released out. If you see the microspore, each microspore is the unicell. If you in tetrad, if you separate the one microspore or pollen grain, which is unicell, and which is made up of two layers, outer one called an exine, inner one called as intine, and one nucleus present. That this is microspore or pollen grain. Later, this one nucleus further divided into further nuclei. And now, if it is single nucleus, this is called microspore or pollen grain. If it developed into more nucleus, now it is called as male gametophyte development. Let's see the number of nuclei in male gametophyte. How it is developed into male gametes. Let's see. Male gametophyte development. Remember, male gametophyte development from the, where it is, from the microspore only. With the male gametophyte development. Right. Just now we discussed about young microspore or pollen grain. Which is young microspore or pollen grain, unicell, exine, intine, nucleus, cytoplasm. That's it. Later, what happened? In this uninucleus, while it is developed into male gametophyte, divide mitotically as a result formation of two nuclei. And these two nuclei again divided in mitotically as a result formation of one, maybe 3 or 4 nuclei. Here the prothallel cell, generative cell, tube cell, 3 cells. Finally developed into 3 cells, or some families 2 cells also formed. And some family mostly gymnosperm 4 cells also formed, but mostly 3 celled or 2 celled. Remember, here in diagram we can see the 3 celled stage of male gametophyte development. Right? If it is one nucleus, it is microspore or pollen grain. When it is developed into 3 nuclei, that is called as a male gametophyte. What happened actually? This after, let me try here. 2 nuclei form. Again, these 2 nuclei again divided mitotically. 4 nuclei form. One is prothallel cell, second generative cell, and third tube cell. Another cell also formed that it can destroy. Only 3 major or 2 major. So anyway, 3 cell stage formed. In this first one, the small one called as prothallel, here prothallel, and the middle one it is called generative cell, and last one tube cell. The function of tube cell, which forms the tube, it forms the tube like this tube. After formation this pollen tube, the what happened? This generative cell nucleus reached the tube. It is tube nucleus. Later, this generative nucleus divided into 2 male gametes. Now the 2 male gametes present inside the pollen tube. Anyway, 2 male gametes also formed. Right? So this is the male gametophyte development. Clear? These diagrams are very, very important. These diagrams very, very important to development of male gametophyte. Which is, so that means You can write in examination point of view based on this diagram only, from microsporogenesis to male gamete formation. Is it clear? Now, now the next one, megasporogenesis and female gametophyte development. Here also same, the megasporogenesis is the short answer. Female gametophyte, very important, or female gametophyte or embryo sac, it is very important for long answer. In female gametophyte embryo sac, there are three types: monosporic, bisporic, tetrasporic. We will discuss later. The first, let's take the megasporogenesis slide. Just wait. Right. Let's see megasporogenesis. Before that, you should know about the ovule or megasporangium structure, as we discussed earlier in embryology introduction. So first of all, let me draw a rough diagram about the ovule. Ovule. For example, rough diagram. Ovule wall. This is micropyle. And here somewhere here the funicle. So this is the ovule structure. What are present inside? Only in initial stage, only two parts. One is integument. This is integument. And another one is, where is blue color? Nucellus cells. The blue color nucellus cells. Only here the two parts in ovule or megasporangium. Right? So these nucellular cells, one of the nucellular cell inside the ovule further developed into archisporial cell. And this is further developed into megaspore mother cell, enlarged in size, which is diploid in state. Megaspore mother cell, diploid in state. So after formation, this megaspore mother cell here, this is megaspore mother cell, megaspore mother cell. Like a microspore mother cell, here also say megaspore mother cells are formed inside the nucellular cells, inside the ovule integument. So after megaspore formation, megaspore mother cell formation, in megaspore mother cell, the meiosis division takes place. So here the first meiosis one, as a result, formation of two nuclei. Second meiosis, as a result, formation of four nuclei. Let me draw here clearly. Megaspore mother cell, meiosis 1. After meiosis 1, two nuclei formed. Meiosis 2, four nuclei formed. Like this. This process called as megasporogenesis. Is it clear? This process called as megasporogenesis. After formation megaspores, these megaspore further developed into female gametophyte or embryo sac. So this is the whole concept of megasporogenesis. Let's discuss here once again. Here the first one, this is megaspore mother cell. Don't get confused here. There is no point to confuse. Megaspore mother cell, meiosis 1, 2 nuclei, meiosis 2, another 2 nuclei, total 4. No, these 4 called as megaspores. After formation megaspore, these megaspores all are haploid in condition. This haploid condition megaspores further developed into embryo sac or female gametophyte. This structure called as female gametophyte. Now let's discuss about the female gametophyte development. The female gametophyte development very important for long answer. Megasporogenesis for short answer. Let's discuss female gametophyte development or embryo sac development. In this, there are three types. One is monosporic development. Second one is bisporic development. Third one, tetrasporic development. You don't get confused here. The simple mono means single. In monosporic development, after formation four spores, let's take another one. Here the 1, 2, 3, 4. Out of 4, 3 degenerate, only one active. This only one megaspore involved in development of female gametophyte. That is called monosporic. If we take bisporic condition, in bisporic, the same way, 2n megaspore mother cell, first meiosis 2, maybe here the one nuclear degenerate, and again another meiosis division takes place 2. As a result, this one becomes 2. Already one degenerate. Now these 2 megaspore involved in formation of female gametophyte. That is bisporic condition. You can assume what is tetrasporic condition. In tetrasporic condition, after formation of 4 megaspore, each and every megaspore involved to development of the embryo sac or female gametophyte. Let's see one by one. The first one, monosporic development. In your diagram already given, this type is monosporic development. And clearly we can see later. We can take one by one. So anyway, before that, after finally when the formation of female gametophyte embryo sac, here in Chalaza side end, 3 antipodal cells are formed, micropyle end, where the 2 synergids and female gamete, egg or female gamete develop. Either side synergids are developed, and in middle polar nuclei formed. So the total number of cells in embryo sac female gametophyte 1, 2, 3, 4, 5, 6 and 7 cells. But in terms of number of nuclei 8, because in polar nuclei, one cell, polar cell, it has the 2 nuclei. That's why the number of cells in embryo sac 7 and number of nuclei 8. Right? So this is the megasporogenesis. Now let's discuss about the monosporic development, later bisporic and tetrasporic final. So first one, let's see the monosporic development. In monosporic development, let me take the PPT slide here. Just wait. Let's wait. Image. Monosporic, bisporic, tetrasporic. Helium type. Okay, right. This also not one. Just wait. Where is the proper one? Right. So if we say this type of female gametophyte, first discuss one by one. First two, monosporic development. After that, bisporic development. Let's see here these two, monosporic, and next to bisporic. This is bisporic, and remaining all are tetrasporic. First, let's see one by one monosporic development. In monosporic development, let's see the megasporogenesis. This is megaspore mother cell. You can here clearly given the meiosis 1. As a result, it is 2 nuclei formed. Meiosis 2, 4 nuclei formed. We can observe after formation 4 nuclei at the side of the micropylar side. Here, 3 nuclei degenerate. Only one remain. This one nuclei in further divided mitotic division, another mitotic division, final third mitotic division, which finally forms embryo sac. And if you take the another monosporic, it is 4 nucleated. In monosporic, 8 nucleated polygonum type and 4 nucleated enotherotype. In enotherotype, the same megaspore mother cell, first meiotic division 2, second meiotic division 4. After formation, we can see here the chalaza side, micropylar up, below side, chalaza side. Here, the chalaza side 3 degenerate, only one active. This only one divide mitotically, 2 nuclei formed, again divide mitotically, only 4 nuclei formed. That's it. So this is called monosporic condition. Let me draw clearly monosporic development here. In monosporic development, clearly, let me draw. Right. First of all, megaspore mother cell diploid. First meiotic division, 2, second meiotic division, 1, 2, 3, 4. What happened? 3 micropylar degenerate, only one active. This only one nuclei divide mitotically. After meiotic, it follows mitotic only. After mitotic, what happened? 2 nuclei form. First mitotic, empty mitotic, remember it. And Second mitotic, 4 nuclei formed. And third mitotic, 1, 2, 3, 4. Here 4, 1, 2, 3, 4. 4 formed. Later what happened? During formation embryo sac, at micropylar side 4, 1 migrate toward the center. Chalaja side 4, 1 migrate toward the center. How many remains? Upper side remaining 3, 1, 2, 3. And Chalaja side also remain 3, 1, 2, 3. Later they are covered by walls and they are developed into cells. So this is embryo sac development. This is polygonal type embryo sac. This is most probably appeared in angiosperms. So anyway, in this is egg or female gamete. The either side cells, these are synergids. And the center polar nuclei. And these are antipodals. The Chalaja side antipodals. Now this is polygonum type. After formation polygonum type, and another type is formed in monosporic, that is Enothera. What happened in Enothera? Let's delete one by one here. In Enothera, just wait. In Enothera, which is also monosporic type, same, sorry. Let me take whiteboard. Sorry. Yes. Same megaspore mother cell. First meiotic, remember, don't get confused with two words, meiotic and meiosis. First meiotic and mitotic. First meiosis, the same like a polygonum 2. Second meiosis, the same 4 formed. But in polygonal type, the side of the micropyle side 3 degenerate. But in Enothera, Chalaja and Chalaja side, below side 3 degenerate, only 1 active. Now this one enter into the mitotic only. First mitotic division as a result formation of first mitotic formation 2 nuclei. And again, another mitotic, 4 nuclei formed at the side of micropylar. And there is no third mitotic division. After 2, the finally formed the embryo sac. So anyway, from the micropylar side, one nuclei migrate toward the center. The rest remaining 3 at the micropylar side. These are settled as a Philippum apparatus. In Philippum apparatus, like a first same like polygonum egg, central egg, 2 side, either side, synergid cells. But in center, polar nuclei formed. But in polar nuclei, in polygonal type, 2 nuclei present. But in polar nuclei, how many nucleus present in Enothera type? Only 1. What about the antipodal cells? No antipodal in Enothera. Because in this Hinothera, there is no the third meta-division. It is divided until the formation of 4 nuclei only. It utilizes the only 2 mitotic divisions. So this is called as Hinothera type. And next one, bisporic. If you see bisporic type, let's take the diagram. Sorry. So this is bisporic, Lem type. Another one is Endemian type. The same, both are mostly same. That is first megaspore mother cell 2n. First meiotic division, first meiotic here given. First meiotic division, 2 nuclei formed. Second meiotic division, actually 4 nuclei formed. 4 nuclei after formation 1, 2, 3, 4. The side of micropylar. Micropylar side 2 degenerate. Only how many remain here? 2 remain. And these 2 megaspore involved in development of the female gametophyte. That's why it is considered as a bisporic development. So these 2 megaspore, the first meiotic division, as a result formation of these 2 micropylar side 2 and chalaza side 2, 4 nuclei. Again developed into another mitoid division. The finally the micropylar side 4 and chalaza side 4 formed. After formation, either side 4 nuclei, one of the either side 1 nuclei migrate toward the center. As a result, formation of polar nuclei, and as it migrate from either side, the rest of the remaining either side only 3 nuclei. The upper micropylar 3 nuclei surrounded by cell wall, and this chalaza side as well surrounded by cell wall. As a result, finally it's developed into female gametophyte. So filiform operator micropylar side egg and synergid either side, and chalaza side antipodals 3, and central as it is. No changes the secondary nucleus. This is Lem type. And another type is bisporic type Endemian. Endemian also same process, only one change. That is column 3, 1, 2, 3. In the 3 after formation 4 nuclei, the destroying the 2 nuclei from the chalaza side. The destroying of the 2 nuclei after formation 4 nuclei after 4, 2 degenerate from chalaza side, that is Endemian type. From micropylar side, that is Lem type. But end product commonly common. That is the polygonum. That is the embryo sac formation. 7 cells, 8 nucleus, no change at all. So this is bisporic type. And tetrasporic type, here the given peperomia, pinia, drusa, fritillaria, plumbagella, plumbago. They are all tetrasporic type. In tetrasporic, as I said earlier, what happened? In this, first let's see the peperomia type. In peperomia, this is megaspore mother cell, first meiotic division, second meiotic division after formation 4 nuclei, 4 megaspore. This 4 megaspore utilized in formation of embryo sac. This 4 further divided into mitotically. Total 1, 2, 3, 4, 8 nuclei formed, and again further divided into another mitoid division, 8 into 2, 16 nuclei formed. After formation 16 nuclei, here the after formation embryo sac from either side, that means 4 sides, micropylar, chalaza, and lateral side, 2 nuclei migrate towards the center. As the 2 nuclei migrate towards the center, and the each corner only remain 2 nuclei only. As a result, if we take the filiform operator, 1 synergid present and 1 egg cell present, but there is no further another synergid cells. And if we take the center cells, that means secondary polar nuclei, how many nuclei present in polar cell? Secondary cell. In secondary cell, as it the 2 migrate toward the center, 4 into 2, 8 present inside the secondary nuclei. Then rest of the all considered as the antipodal. How many? Here the 8 2 10 the remaining 6 antipodals. That is peperomia type. The same that way. The all 16 Peony also same, Druce also same, but Fritillaria 8-celled nucleus, Plumbagella 4 nucleus, Plumbago 8 nucleus, tetrasporic typing Adaxa, that is 8 nucleus. So these are the development of embryo sac. Now, so this is female gametophyte development. We will discuss further later. Tomorrow I will complete the third unit also. Just see this chart.